Hello everyone, welcome to Latest Trends 2023, the channel where we explore the most exciting and innovative trends of the future. I'm Jay, and today we're going to talk about gene editing, a technology that can literally rewrite the code of life. What is gene editing? Gene editing is a type of genetic engineering that allows us to make precise changes in the DNA sequence of a living organism, essentially customizing its genetic makeup. DNA is the molecule that stores all the information needed for life, and it is composed of 4 bases: adenine, A, thymine, T, cytosine, C, and guanine, G. These bases pair up to form the famous double helix structure of DNA, and the order of these bases determines the traits and functions of every cell in our body. Sometimes, however, there are errors or mutations in the DNA sequence that can cause diseases or undesirable traits. For example, sickle cell anemia is caused by a single change in the DNA base from A to T in the gene that codes for hemoglobin, the protein that carries oxygen in our blood. This change makes the hemoglobin molecules stick together and form sickle-shaped red blood cells that can clog blood vessels and cause pain, infections, and organ damage. Gene editing can potentially fix these errors by cutting out the faulty DNA and replacing it with the correct one. This way, we can correct genetic diseases or enhance desirable traits in plants, animals, or humans. How does gene editing work? Gene editing works by using special enzymes called nucleases that can cut DNA at specific locations. These nucleases are like molecular scissors that can recognize and bind to a target DNA sequence and make a double-strand break in the DNA strands. Once the DNA is cut, it can be repaired by 2 main mechanisms: non-homologous end joining, NHEJ, or homologous recombination, HR. NHEJ is a process where the broken ends of the DNA are simply joined back together by other enzymes. This can result in insertions or deletions of some bases at the cut site, which can disrupt or disable the function of the target gene. This is useful if we want to knock out a gene that causes a disease or an unwanted trait. OUR is a process where the broken ends of the DNA are repaired using a template DNA molecule that has the desired sequence. This template can be provided by another copy of the same gene from another chromosome or by an artificial DNA molecule that we design and introduce into the cell. This way, we can insert or replace a specific DNA sequence at the target site. This is useful if we want to correct a mutation or introduce a new trait into a gene. What are some examples of gene editing? Gene editing has been used for various purposes in different fields of science and technology. Here are some examples. In medicine, gene editing has been used to create animal models of human diseases for research purposes. For example, researchers have used gene editing to create mice with muscular dystrophy, cystic fibrosis, or Alzheimer's disease. These models help us understand how these diseases work and test potential treatments for them. Gene editing has also been used to modify human cells for therapeutic purposes. For example, researchers have used gene editing to correct the mutation that causes sickle cell anemia in human blood stem cells. These cells can then be transplanted back into patients to produce healthy red blood cells and cure them of their disease. In agriculture, gene editing has been used to improve crops and livestock for food production. For example, researchers have used gene editing to create rice plants that are resistant to drought or salt stress, or pigs that are resistant to a virus that causes porcine reproductive and respiratory syndrome. In biotechnology, gene editing has been used to create novel products and applications. For example, researchers have used gene editing to create bacteria that can produce biofuels, insulin, or biodegradable plastics. They have also used gene editing to create animal organs that are compatible with human transplantation, or animals that can glow in the dark. What is CRISPR-Cas9? CRISPR-Cas9 is a gene editing tool that has revolutionized the field of genome engineering. It is based on a natural system that bacteria use to defend themselves against viruses. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which are segments of DNA that contain repeated sequences separated by unique spacers. These spacers are derived from the DNA of viruses that have previously infected the bacteria. Cas9 stands for CRISPR associated protein 9, which is an enzyme that can cut DNA. How does CRISPR-Cas9 work? CRISPR-Cas9 works by using a guide RNA molecule that is complementary to a target DNA sequence. The guide RNA binds to the target DNA and directs the Cas9 enzyme to cut the DNA at that location. The cut DNA can then be repaired by NHEJ or HR, resulting in gene editing. What are the advantages of CRISPR-Cas9? CRISPR-Cas9 has several advantages over other gene editing tools. It is simple. It only requires a guide RNA and a Cas9 enzyme to perform gene editing. Versatile. It can target almost any DNA sequence in any organism. Efficient. It can edit multiple genes at once or create large deletions or insertions. Cheap. It is relatively inexpensive and accessible compared to other gene editing tools. What are the challenges and risks of CRISPR-Cas9? CRISPR-Cas9 also has some challenges and risks that need to be addressed before it can be widely used for clinical or commercial purposes. These include off-target effects. CRISPR-Cas9 may sometimes cut DNA at unintended locations that are similar to the target sequence, causing unwanted mutations or damage to other genes. Mosaicism. CRISPR-Cas9 may not edit all the cells in an organism or tissue uniformly, resulting in a mixture of edited and unedited cells. This can reduce the effectiveness or consistency of gene editing. Immune response. CRISPR-Cas9 may trigger an immune reaction in some organisms or individuals, especially if the Cas9 enzyme is derived from a foreign source. This can cause inflammation, rejection, or allergic reactions. Ethical issues. CRISPR-Cas9 raises ethical questions about the safety, consent, and equity of gene editing, especially when it comes to human germline editing. What are the ethical issues of gene editing? Gene editing poses ethical dilemmas that require careful consideration and public debate. Some of these issues are: safety. Gene editing must be proven to be safe before it can be offered as a treatment option. There is a risk of off-target effects, mosaicism, immune response, or unforeseen consequences that could harm the health or well-being of individuals or populations. Consent. Gene editing must respect the autonomy and dignity of individuals who undergo or are affected by gene editing. There must be informed consent from those who receive gene editing or donate their cells or tissues for gene editing research. There must also be respect for the rights and interests of future generations who may inherit edited genes. Equity. Gene editing must be accessible and affordable for those who need it and not create or exacerbate social inequalities or injustices. There must be fair distribution of benefits and risks of gene editing among different groups and regions. There must also be protection from discrimination or exploitation based on genetic traits or modifications. Conclusion: gene editing is a powerful technology that can change our lives for better or worse. It has great potential for improving human health and society, but also poses significant challenges and risks that need to be addressed responsibly and ethically. CRISPR-Cas9 is a simple, versatile, efficient, and cheap tool that has made gene editing easier and faster than ever before, but also raises new ethical questions and concerns. As we enter a new era of genome engineering, we need to be aware of the possibilities and limitations of gene editing, and engage in public dialogue and deliberation about its implications and governance. Thank you for watching this video. If you liked it, please give it a thumbs up and subscribe to our channel for more videos on latest trends 2023.